So in order to see something that is far away or close up, we have to change the shape of the lens, and we call that accommodation. We said if we want to focus on something that's far away, these ciliary muscles are going to relax. That changes the shape of the lens and makes it more flattened out, so we can see things that are far away. If we want to see something that's close, then those ciliary muscles are going to contract, and this is going to make the lens more oval in shape, more rounded. And then that helps us to focus on something that is close. Now there are terms, and we said we did need to talk about some of the eye disorders and then move on to hearing. Well, if we are not able to see something that is, or if we can only see things that are close, nearsightedness, okay, that's called myopia. Hyperopia is when you can't see things that are close and you can see things that are far away. These can be corrected with different lenses. Go to your eye doctor, ophthalmologist. Okay. Astigmatism. Astigmatism is when the cornea or the lens is not smooth. Maybe it's curved, or something, maybe it's injured, maybe you have a scratch, something like that. It's not smooth and things look blurry. Okay, so it's hard to focus. Now, what is glaucoma? Has anyone heard of glaucoma and may know what maybe some things that are related to glaucoma, or what can cause glaucoma in a person as we age?
Is it like a cloudy film that accumulates?
That would be a cataract. If you have like a film or cloudiness, that would be a cataract. The proteins are changing. Glaucoma. Has anyone heard of glaucoma? Well, I'm gonna come back to light adaptation, because we did get that far. Okay. So glaucoma is when you have too much fluid in the eye. This causes increased pressure in the eye, or intraocular pressure increases. So think about where the lacrimal bone is. We have our opening for our tear duct, and it goes into the nasolacrimal pathway to drain, so that a lot of times when people are crying, their nose is running because it's draining. Sometimes that duct can be clogged up. That can increase intraocular pressure if we're not able to release the fluid. And then sometimes there's too much secretions. And that can cause too much buildup of fluid in the eye. This can be something that occurs in people with diabetes. Okay. The cataract or clouding of the lens is what you were talking about, Patricia, and there's surgery, laser surgery, and things like that that can remove that layer or that area that has been damaged, or where the proteins are actually starting to lose their shape and make it cloudy and not clear. Right. Conjunctivitis, inflammation of the conjunctiva, pink eye, we talked about that. We mentioned astigmatism just now, and those are the major ones. Color blindness is genetically influenced, and we'll be able to test that in laboratory. All right, going back briefly to the picture of the rhodopsin cycle. So this is what we also tried to draw out, well, not as much detail, but when we are exposed to light, okay, the rods on the retina protein, they're called rhodopsin. Okay. When light hits rhodopsin, it's going to change shape. Here we see that the opsin comes apart or is not now in the same formation with retinol, so they change. And this activates G protein coupled receptors that are on the inside of the membrane to close the sodium channel. So we said today, if sodium channels close, this is going to make the membrane more negative, so we have hyperpolarization. We can put things back in place if we have energy and ATP. And this will occur when we are adapted to the new setting, or maybe a lighter area, a lighter room, or when we are back in the dark. They also give you a chart that actually shows you this process, and it shows you the membrane potential. And just remember, if we hyperpolarize, that means we become more negative. Closing those sodium channels when we're exposed to light will make the membrane more negative, hyperpolarized. Questions about that summary or recap on the eye, structures, light adaptation, any of those terms? Just be aware also that we do have dark adaptation. Remember, the opposite happens if we're in the dark, if we're in light and then we go into a dark setting, then the pupils will dilate. Then we will activate the rods and try to get as much light as possible. Okay.
So we depolarize
In the dark.
and we depolarize in the dark, and the sodium channels will open.
Yes. In the dark, we will have depolarization and opening of those sodium channels on the rods. This picture is also just showing us rods actually look like a cylinder, and the cones kind of look like a little triangle cone. There's not a lot that I want you to know about the cones, except that they detect color, and the three primary colors that they do detect are red, blue, and green. Okay. Red, blue, and green. Okay. So the three primary colors on the UV spectrum that we can see detected by the cones would be red, blue, and green. It's a different protein there. It's called iodopsin, just so that you don't get that mixed up with rhodopsin from the rods. So our last objective for chapter 15 is to review the structures of the ear and outline hearing conduction and balance. How are we able to perceive those senses? Okay. This is just interesting information too, just to be aware that when we see something from our eye, it does have to cross over to the opposite side of the brain to get to the occipital. So this is just showing us that we have different visual fields. We have a nasal or medial field, and we have a peripheral or temporal field. And what we can see from the nasal field is going to cross over to the opposite side of the brain at the optic chiasm that you guys labeled in the sheep brain. The only thing to also mention that's important about that, the pituitary gland, remember the pituitary gland is sitting right here. So sometimes if people have an enlarged pituitary or pressure right there at the optic chiasm because of an enlarged pituitary, it can cause vision problems. And this is also where we get some of those visual reflexes with the superior colliculus. All right, so here's our primary primary diagram for the ear anatomy, okay? So before the final lab exam, we have to be pretty firm on that eye anatomy and pretty firm on that ear anatomy. We won't have a quiz, so that's why you were supposed to do the labeling and connect. So we have an external or outer ear. This includes your auricle or pinna and the ear lobule. Here, this area here coming in is the eustachian tube, or you can remember the bony structure, external acoustic or auditory meatus, okay? Or not eustachian tube, but the eustachian canal, okay? The eustachian tube or auditory tube is actually here that goes and connects to the middle ear. So the middle ear includes your eardrum, which is called anatomically the tympanic membrane. Please don't put eardrum on your test next week. Tympanic membrane. And then you have 3 ear bones. MIS, in that order. Malleus, incus, and stapes. We'll see a close-up here at the bottom. MIS. So if you've ever played the drums, or you've watched someone else, or listened, or been observant, you know, if they're hitting that drum and you hear it, you know, the little metal pedal is actually hitting it, causing that sound. That's like sound waves coming into our ear. It's going to hit that first tympanic membrane and then vibrate through the ear bones. It's going to vibrate through these 3 until it hits the opening to the inner ear, and that is referred to as the oval window. Those vibrations go through, so you may have like a sequence question. Put these in the right order for which sound waves or vibrations will go through to the ear. We're trying to get to these nerves back here so we can transmit this sound to the brain. And so you have malleus, incus, and stapes in the middle ear, and then we get to the oval window, and that sound wave can go into the inner ear. There are 3 major structures in the inner ear: semicircular canals, the vestibule, and these 2 are going to be important for balance. The vestibule and the semicircular canals are there for balance, so it's this kind of area here. Okay. The semicircular canals we're going to see also give us like a 3D spatial arrangement. Some are up, a branch is over, and a branch is kind of lower. So you kind of know where you are in space. That's your, you know, balance. Warsaw, can you guys still see me?
Yes.
Okay. I didn't want to just make a whole bunch of signals and you guys can't see. All right. So anyway, cochlea is going to be the one for hearing. So this is the one for hearing. So vestibulocochlear nerve is a combination of a cochlear branch for hearing. Here you see the cochlear nerve to the cochlea, kind of looks like a snail shell. And then the vestibular branch here going to the vestibule and the semicircular canals.